Good day, everyone. This is Dr. Shama Khan from Wits University Vaccines and Infectious Diseases Analytics Unit. I'm working as a bioinformatics researcher, and today I will be presenting one of my projects that is Fabl, which is a druggable target in identification of novel inhibitors against Klebsiella pneumonia. So why we choose this Fabl protein because and also for Klebsiella pneumonia because this Klebsiella pneumonia is a gram-negative bacteria which is like emerging opportunistic pathogen causing life-threatening infections to human and animal microbiome. This bacteria, it belongs to Enterobacteriaceae family with the fifth leading cause of pneumonia, mostly in newborn units, intensive care units, and individuals with compromised immune system. So my study, it offers the identification of potential inhibitors of this Fabl protein of Klebsiella pneumonia to overcome the burden of antimicrobial resistance in Klebsiella pneumonia therapeutics using advanced molecular modeling and bioinformatics approach. So the reason behind choosing this Fabl protein is because it plays a key role in fatty acid biosynthesis and is considered now like it's a recently novel discovered attractive druggable target. And for this, we use all, as I said, molecular modeling approaches, and we have identified out of 300 compounds, we have identified top 3 leads, which could be the potential inhibitors by using integrated drug discovery approach and other molecular modeling techniques. So as you can see in this slide, we this is on the left hand side, this is like the Fabl protein, and which is having a known inhibitor, that is traclosan drug. It is a known inhibitor of Fabl protein, which is sitting in the binding site, and NADH. NADH is it contributes. It is a cofactor and it contributes in the formation of a stable complex with the triclosan drug at the enol substrate site. Hence, it was important to keep this cofactor. So we performed all the studies using this cofactor. And the structure of the Fabl was not known, so we did apply homology modeling approach. We built the three-dimensional structure from the FASTA sequences, and then we docked the known inhibitor into the known binding site, and this is the structural representation of the binding site of Fabl drug target with triclosan and an ADH cofactor. Afterwards, we built the homology model. We did dock triclosan in the known binding site. We generated the E-pharmacophore model based on the receptor ligand complex and the other binding site residues. As you can see on the left hand side, this is like the hydrogen bond acceptor, hydrogen bond donor, green and orange rings, and then the benzene rings. And on the right hand side, these are in the blue color, these are the binding site residues. These are the top 3 structures. What we did, as I said, it is like the integrated drug discovery approach. So we generated the homology model. After that, we docked the known inhibitor into the binding site along with the cofactor NADH, and then we generated the E-pharmacophore model, and then we screened the database against the known inhibitor, that is triclosan. We used the PubChem database with 95% similarity search, and we have got around 300 compounds. And after performing molecular docking based virtual screening, we got 36 compounds based on the standard precision module implemented in Schrodinger. Afterwards, we got 23 compounds based on the extra standard precision module. And then we got, we put the parameter that the binding energies, which is between -8.5 to -10, we got top three compounds, that is compound 1, 2, and 3, and along with the control inhibitor. So the binding energy was of these three compounds were better than the control inhibitor. The binding energy of the control inhibitor was minus 8.45, and these three compounds were having minus 10.01, minus 8.95, and similarly compound 3 also shows minus 8.79. This is the three-dimensional structure of and the two-dimensional structure of the Fabl protein in complex with triclosan. On the left-hand side, you can see the two-dimensional maps, and on the right-hand side, the same, it is the orientation in the 3D form. And this is forming tyrosine 119 was the key contributing residues. It formed pi-pi interaction with the pi-pi stacking with the receptor. So this is like the two-dimensional or interaction map, and the same is the three-dimensional orientation of the triclosan with its binding pose in the cavity of Fabl protein. Afterwards, we did dock like these top three compounds, and as you can see, tyrosine 119, tyrosine 129, these residues, and alanine 68, they all contributed in the binding of these three compounds to Fabl protein, and they form like hydrogen bond interaction, pi-pi interaction, and halogen bond interactions. Then what we did, we took the studies to next level, and then we extended our studies and we performed the structural dynamics of Fabl in complex with the other top three compounds and the control inhibitor. So as you can see, the most fluctuating or most like deviating compound inhibitor was like the non-inhibitor in purple color. On the left hand side, this is the root mean square deviations, and then on the right hand side, this is the root mean fluctuations. So root mean deviation is the compact, it shows the compactness of the protein only in the linear chain, and then you can see the same amino acid residues on the right hand side. They are like highly fluctuating, and especially in the triclosan, that is the known inhibitor, but other compounds are good. So that's how we came to conclusion that our compounds, the screen compounds, are better than the known inhibitor. Same pattern followed in the radius of gyration and SASA, that is solvent accessible surface area. So afterwards, like from all these four, these are the four basic structural dynamics. So we concluded based on these ones that our compounds are better than the known inhibitor. Afterwards, we performed some of like secondary structure analysis, like what changes happened when we docked our known inhibitors into the active site, and we found that the amino acid, because the amino acid sequence is arranged in the form of polypeptide chains, and later we consider it as a secondary structure of proteins. So this secondary structure falls into a three-dimensional structure to form a tertiary structure of the protein, and it was necessary to observe the changes associated with the tertiary structure after binding of these three ligands or inhibitors in terms of alpha helix, beta strand, 310 helix, turn, bend, and others as shown in this figure. So A is the control drug, B is the compound 1, and the differences are visualized like that there are like more bends and more turns in compound 1 in comparison with the known inhibitor. Same you can see in compound 2 and 3 that there are the bends are more, like the red color indicates the bends and then the green color indicates the pi. So we can see here that these alpha helix, beta strands, turns, and bends, they are more dominating in the top three screened compounds. Further, we did the principal component analysis for all the top three leads and with the known inhibitors. So this PCA is an essential parameter which identify the conformational deviations in the structure of protein to explain the movements of residues after the ligands bind to the binding cavity or the binding site. This analysis, it produced extremely correlated and anti-correlated fluctuations originated from the simulation trajectories. So as you can see from x-axis eigenvector A versus eigenvector 2, the more positive relation can be seen in the green color, that is compound 1, and following compound 2, 3, and afterwards there is like the negative correlation of the residues or negative movement between the monocytic residues in the non-inhibitor, that is triclosan in black color. So all of this analysis, and we performed 200 nanosecond of molecular dynamics simulation, and this paper is going to be published soon. These are the preliminary results, and afterwards, because of these like top three compounds, so compound 1 was the most dominating. So this study could help like overcoming the MDR, that is multi-drug resistant and extremely drug resistant, which is increasing at alarming rate worldwide, more than and specifically in the developing countries like our country, like especially African continent, with the high prevalence of antimicrobial resistant pathogens like Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, that is all the escape pathogens. And these pathogens, they cause life-threatening infections in infants by affecting adults and infants by affecting essential organs like liver, brain, bladder, bloodstream, and lungs, etc. So because this approach, it's cost efficient, economical, and productive strategy. So we screen the compounds virtually using high throughput virtual screening, following the lead optimization protocols with integrated in silico techniques to produce promising compounds. So in this study, as I said, we use the integrated computer-aided drug discovery approach to screen the lead hits from a chemical database, that is PubChem, based on the known inhibitor scaffold to overcome the growing threat of antimicrobial resistance. Thank you so much, audience, for listening to me, for giving your valuable time. And this is, I would like to acknowledge, this is on the left-hand side is Professor Shabir Madhi. He's my host, supervisor, and my boss, of course, and then on the right-hand side, it's my group. I would also like to thank Dr. Courtney P. Olvajen and Dr. Vicky L. Belli. They are also my supervisors. I'm working with them, not exactly the supervisor, but my host. And I would like to thank Vida for providing me the platform to do this kind of research. And my funding is from NRF Sachi. I would like to thank Wits University, URC as well, for funding for my project. And thank you so much for listening to me, audience. Have a good day.